In this presentation, I will introduce the concept of a haplotype. Haplotypes are a key idea in genetics and genomics, helping us understand how genetic variations are inherited and how they relate to traits and diseases. A SNP is a one-letter difference in the DNA sequence. For example, one person may have an A at a certain spot, while another has a T. These small differences play a role in creating the variation we see between individuals. When SNPs are close together on a chromosome, they act like a small block. Even when recombination occurs, these nearby SNPs are less likely to be separated. So they often get passed down together as a set. When several nearby SNPs line up on a chromosome, their order forms a kind of pattern. For example, one person may have ATC, while another has ACT. These different patterns of SNPs are what we call haplotypes. A haplotype is the combination of several SNP alleles that lie close together on a chromosome. Because recombination rarely splits them apart, they tend to be inherited as one block. So the entire pattern of those SNPs represents a person's haplotype. On this slide, I explain the difference between a genotype and a haplotype. A genotype refers to the pair of alleles an individual has at a specific genetic position, for example, AA, AG, or GG. It focuses on variation at a single site and does not tell us how nearby variants are arranged. A haplotype, on the other hand, represents a set of linked variants located on the same chromosome. It preserves the phase information, meaning it shows which alleles are inherited together. In short, a genotype tells you what alleles you have, while a haplotype tells you how those alleles are arranged on your chromosome.
I will explain the types of haplotypes. Examples include HLA haplotypes, ApoE haplotypes, mitochondrial DNA haplogroups, and Y-DNA haplogroups. Regarding HLA haplotypes, the HLA genes are divided into class 1 and class 2, and haplotypes are formed by combinations of these genes. A person inherits one set of haplotypes from their father and another from their mother, so each individual carries two sets. The probability of HLA matching between siblings is 25%.
MHC haplotypes play a critical role in organ transplantation. When the HLA differences between a donor and a recipient are large, the immune system is more likely to recognize the transplanted organ as foreign, which greatly increases the risk of rejection. Even small mismatches in HLA can influence immune reactions, but larger differences create strong immune activation. This is why close HLA matching is essential for successful transplantation and long-term graft survival. Different MHC haplotypes affect how efficiently the immune system recognizes pathogens. Some haplotypes make individuals more resistant or more vulnerable to specific infections, Additionally, certain MHC combinations are linked to autoimmune diseases, such as type 1 diabetes and rheumatoid arthritis. MHC genes are among the most diverse genes in the human genome. Because of this diversity, they are valuable markers for studying population genetics, migration patterns, and human evolutionary history. Overall, MHC haplotypes form a crucial link between genetics and immunology. They influence disease risk, transplantation outcomes, and population health, making them essential for both medical practice and biological research.